Welcome to the Machines and Molecules podcast. Machines and Molecules hosts guests from chemistry, biochemistry, and life sciences. And our guest today is Stas Mazurenko, who is a team leader at Loschmidt Laboratories in the Czech Republic, a lab that is very well known for machine learning in protein design and protein optimization. Welcome, Stas.
Thank you for having me here. Hi, Ingvar. And hello, everyone. Yes, great. Thank you.
Yeah. So at the moment, you are quite busy writing grants. You've told me before we started recording. When is the deadline approaching? How stressed are you?
A little bit stressed. I'm primarily stressed when I know that I depend on other people. And of course, usually, like when you write grants, it's not only you, but you also need to run it by by the university people, your collaborators, etc. So I also try to finish it earlier. And this is exactly what stresses me out most commonly. But then, like when we are about to submit, usually it's all on the good track. I hope I won't jinx it this time.
Okay. So Loschmidt Labs is known for machine learning in protein design. Actually, I think it was known for protein design even before taking on the machine learning part. You lead a team of 8 people. Maybe I think your background is in mathematics. Is that right?
That's right. Yes.
Yeah. So maybe can you explain to a person from machine learning mathematics what protein design is and why it's important at all?
Yeah. Okay. So I will start a little bit far away, like from the proteins. Yeah. So in general, the focus of our research is are proteins, and proteins are pretty huge biomolecules that you can find in any living organisms, and they are primarily like all those machines that perform all the work. So like for the cells, they are responsible for replicating cells. They are responsible for transport through membranes within the cells. They're responsible for converting some substrates into products, like to make some useful compounds for a cell to survive, etc., etc. So there's like all this machinery of life depends vitally on proteins, and this is why they are super interesting to study, because of course we want to understand how living organisms function, but also we want to understand if we could borrow something from the nature and then use it maybe in the industrial applications. Yeah, for instance, some enzymes that the cells use to produce some metabolites for the cells, we can then maybe borrow and use it to degrade plastic or capture CO2 from the atmosphere, like all those big global challenges. So the problem with proteins comes from the point of view of how many there are. Because typically they are encoded by like a sequence of amino acids, and it can be anything from short peptides, so it will be just like a dozen of amino acids, to like thousands of amino acids, usually connected in really long chains. And all those chains are folded somehow, and then this particular shape of the protein eventually leads to a particular function. And yeah, current estimates are that like in human cells, around 100,000 proteins are being produced. So that's like a huge number. And of course, we would like to understand what they do. But protein engineering, in particular, is interested in understanding if we change those sequences of amino acids forming proteins, what effect it will have on the function of a protein, for instance. And then it turns out to be like quite a difficult task, but very important one, because we know from biology that even a single mutation, which means that just a substitution of one amino acid for another in this long sequence, can completely destroy the function of a protein. And this might cause some diseases, for instance, in humans. And then we also know that some mutations, they can really improve the proteins. And sometimes enzymes in nature have not been fully optimized, because cells have to solve many different tasks. But for instance, if we want to use them in the industry, we quite often need some really specialized enzymes. So then we don't care much about like maybe all those other tasks. So then maybe we can find some mutations that eventually will produce even better catalysts, or even like more stable enzymes that we can use in like high temperatures or harsh industrial environments, all those things. So then it boils down to the problem of you have a sequence of amino acids that encodes a protein, and then you want to understand if you mutate an amino acid in position X from Y to Z, for instance, what effect it will have on the function. So this is like, in the nutshell, what protein engineers are trying to do, because once you know it, you can then improve your proteins, or you can change them in a way that you want, or you can understand the mechanisms of the diseases, because you understand that, for instance, in some humans, particular mutations might destabilize the protein or make it stick to each other and then like cause some adverse effects.
So maybe to break it down even more, you mentioned enzymes. These are catalyzing chemical reactions in the body, right? So
yes.
They're called biocatalysts also for layman's. And when you said they can cause diseases, for example, like people have encoded these biocatalysts in their genes. And sometimes if the encoding is wrong, then that means a certain reaction can function in their body, and that's why they get sick, right?
Yes,
that is
but
one
it might break some reaction that, like the enzyme that is supposed to perform its functions stops doing it properly, or it can like shift the balance somehow in a way that the cells can't cope with. So there are like in biology, the major challenge is that everything interacts with everything pretty much, because in the cell, like all those molecules, they're constantly like in contact. And this is why there are multiple different ways how like one mutation in a sequence can eventually lead to some really serious effects.
Problems. Yes.
Yeah.
And when you say a sequence, it's really just these amino acids are just stringed up like on a chain, right? That you wear on a necklace and you put in all these different building blocks, but maybe it's also comparable to Lego in a way. You have these 20 Lego blocks, and you can chain them up in one way, and then you get one machine, and you chain them up in another way, you get another machine. I sometimes compare it to a bit like a hieroglyphic machine, and with machine learning, you're trying to understand the principles of this machine, but you have a hard time reading really this machine, and machine learning really helps you to understand the mechanisms better, because our human brains are so bad at doing that.
Yeah, definitely. And it's also that these machines eventually they function on the basis of chemistry and physics. So we sort of more or less know the rules according to which like we should be able to simulate, for instance. Yeah, nowadays we perform a lot of simulations of those enzymes, for instance, with different compounds or like proteins interacting with each other. But the problem is that they are so complex. So maybe like breaking it down to the computer science language, that there are so many degrees of freedom that it's really challenging to somehow approach this problem by just brute force, just seeing all the possible combinations, for instance, or just calculating all the possible interactions, because there are so many of them that the systems don't scale very well. And then like we're struggling. And this is where machine learning can come in handy, because then it can provide some shortcuts that we don't need to do everything ab initio, like really calculating all the possible interactions of all the atoms in every amino acid. And this is like hundreds of thousands of atoms, basically for a protein, that you can somehow find some patterns that are relevant for your particular task at hand.
So what you're talking about, the simulation, that's been very en vogue. I'm not sure, even like before my academic time, I think in the 80s, 90s, maybe, where people really simulate the force fields. But I think especially for proteins, this is problematic, because there's so many atoms in proteins, because the amino acids themselves are made up of several atoms that form just the amino acid building block, and then there's hundreds of amino acids chained up. So I guess that's one of the problems for molecular dynamics for proteins. Is that true?
Yeah, definitely. So there are some like algorithms that would try to do some calculations of energies based on, like, for instance, static structures. That is relatively a simple task, because then you just calculate, you have like all pairs of amino acids that might be like just in thousands, and then you can calculate interactions. However, there you typically cannot sample nicely this like conformational space, and then proteins are highly dynamic. So what people have been doing over the last like few decades is to actually simulate, like start from a static structure and then calculate at each point of time like forces, interactions between different atoms, and in which direction atoms should start moving. And then you just solve the physics equation. Yeah, usually they are like some approximations, some fast equations, or you can, nowadays you can decide on the level of complexity or coarse grain that you want your system to have. And then you just do it like time after time. Yeah, in each time point, you just recalculate what happens to your protein.
Yeah, this coarse graining that you talk about, this is kind of trying to look only at the most important forces and trying to like fast forward this physical simulation, right?
Yes. Or for instance, you can also decide to look at the protein as not a collection of atoms, but a collection of, for instance, just residues. So basically you coarse grain each amino acid, which is composed of multiple items, to just like one unit. Of course, then this comes at a cost that you need to redefine its interactions with other balls like this. And this becomes challenging. On the other hand, you gain in the speed of calculations, because then the system size is reduced significantly. And then you can also make some simplifications about, you can even do it on the level of like secondary structural elements of the protein. So bigger structural chunks. You say, okay, I don't need high resolution of every atom or every residue, how they're wobbling. It's enough for me to understand the major motions of the protein. Yeah. And we have some proteins that like have like really big conformational changes, and those can often be captured by those coarse grain methods.
So all of these methods are very physics informed. Now, can you contrast that with pure statistical methods, maybe?
So I think in the force fields, quite often they use some evaluation of or like approximation of those energies from the data. So it's also in a way some statistical, and now we also have like machine learning based tools for this. So this works a lot. And another thing how you can approximate the dynamics is that if you can collect the data, for instance, from the experiments that just samples this conformational space. Yeah, and from this you typically won't see conformational changes, so you won't see it in like as a video, but more you will have these like statistical ensembles of states from which you of course can also dig out some important information. The major limitation here is that our capacity, although there has been a huge progress in those methods, like experimental methods for collecting those different conformations, it's still quite like limited space, and some of the conformations are difficult to capture or observe. For instance, for enzymes, the most sensitive conformational change is this transition state. So when when you have a substrate that gets into the enzyme and then it binds, and then there is a transitional state that actually overcomes the energy barrier necessary to convert this enzyme into the product. But the time scale for this event is super small. So then it's really difficult to observe it in experiments. And then eventually you need to simulate something. The problem is also that like with the simulations is that quite often if we look at the energy landscape of proteins in general, so if you assume that for each conformation of a protein, you can calculate the energy. So then there are basins, so lows of energy, and then proteins, of course, tend to stick to those low energy conformations. And that's a problem with molecular dynamic simulations, because then when you start simulating, by definition, it will stay in this conformation for a long time because it's an energy minimum. So then a lot of effort in the domain is diverted nowadays to this really pushing the system through over the barriers to some new conformations, new low energy states. And this is also somewhere where machine learning is now being used to somehow, because first of all, you need to identify those promising states at the border of the your local minima, yeah, and then also how to then eventually explore the landscape.
Yeah, I mean, there was a lot of cross-fertilization, I think, between machine learning and molecular system simulation, because the energy landscape that you talk about is more or less exactly a loss landscape as well, if you talk about a purely statistical problem. So we have exactly the same problems, or very, very similar problems, only that often, like if you are looking at optimization-based machine learning, you just try to find the minimum, whereas if you are a Bayesian, you're trying to get all of the different states in this loss landscape or posterior, or the energy landscape if you look at simulations.
Definitely, that's the beauty of mathematics in general, and this like modeling that you learn those methods, and then you realize that there are lots of parallels that you can draw between different domains, and it's not only within one field of study like biology, but even across disciplines, because from the moment you can formulate the problem in a mathematical way, then it's some function that you need to either minimize or, as you mentioned, like sample, evaluate some posterior distribution, etc. So then this is, I think, the beauty of having mathematical background, because then you can see all those parallels and you realize how everything is connected. Yeah,
absolutely. And now we talked a lot about what are proteins, what are the methods in machine learning, from machine learning that people use, and from simulation. Maybe to explain to a biochemist or a chemist, why would they want to use machine learning for their problems? When can it come in handy?
So how I usually, because I teach a course like in AI for exactly biologists and chemists. So how I usually motivate the use of machine learning is that, biology is complicated. As a humanity, we were great at like capturing some more or less straightforward patterns. When you have some like linear relationships, so you plot something versus something, whatever you measure, for instance, and then you see like, it forms a straight line. So maybe there is some linear relationship. So those problems are somehow easy to process, and they are easy to see in your data. However, in biology, quite often you need to consider multiple inputs, multiple channels of information. This can come even at such a simple setup that in your experiments, when you produce proteins, you have a lot of parameters to shuffle. You can pick different host organisms in which you're going to produce. You can shuffle how long you want to, like the cells to first multiply before producing your protein, then switch to making your protein, like all those pH conditions, buffers, solutions, etc., etc. So like all that chemistry that comes in hand with like experiments. Then if you start thinking about all the possibilities, it becomes like really huge problem in terms of dimensionality. And then it might be really difficult to see any patterns there. Primarily because you cannot just plot it. So you cannot just plot a graph and then see like, okay, here is my temperature optima, because you want to shuffle at the same time temperature, pH, and then like all those conditions. And this is where machine learning can help. So I'm motivated more in the first approximation, it's the way how to process your data that you collect in the lab. Why? Because you quite often get overwhelmed by the abundance and by the variability in your data, and you want to make sense out of it. And then machine learning gives you just a tool to understand, are there any codependencies in your data? Are there any interesting patterns that you eventually want to explore, even as not a data scientist, but as a biologist?
I was just thinking about the spurious correlation problem. Like if you have so many dimensions and just few data points, which often happens in biology, if you really care to measure everything that you could measure in principle, but oftentimes one data point is very costly, and then you get strange correlations, which are there by pure chance. Have you observed this at any point or not really?
Yeah, definitely. And I think it's also like a piece of the puzzle that is currently missing in many machine learning pipelines, because primarily machine learning developers come from computer science background. And then statistics, I've asked my students around, they didn't have that many courses in statistics, which is a pity because you work with the data. And then apart from just observing that, okay, there is some pattern, there is some, let's call it correlation, between your variables, you want to push it further and you want to understand first, is it like statistically significant or is it just like by pure chance? And second, what affects what in this part of like in this picture. And then I think it's still in the future of the field. I'm like a little bit, no, I'm like optimistic about it, but it's just, quite often you see that computer science is still lagging behind these like developments in statistical methods. On the other hand, I know that, for instance, like in economics and other domains where it's critical to make correct decisions. Yeah, because in biology, I think it's somehow we are allowed to make mistakes, and typically it's not that you're going to ruin some like country's economy by making a bad decision. But in economics or healthcare, for instance, it's really you want to make right decisions. So you want to make sure that when you compare like a drug, whether it's efficient or not, it's statistically significant. It's not just like a spurious correlation. And those domains are much more advanced when it comes to statistical methods. But machine learning is just penetrating there. But I know that also statisticians are starting to use machine learning methods more and more, because before that, the major workhorse in like statistical analysis is like linear regression, or like you try to find first order effects, and then machine learning potentially can allow you to capture more difficult patterns.
Maybe shifting gears slightly towards the interaction of machine learning with lab equipment. How do you see the interactions with automation in particular?
Yeah, I think it's a really good question and very promising direction, because a lot of lab work can be automated. If you think about what people perform on a daily basis, especially, for instance, this protein production involves quite a set of standardized steps that you need to execute. And then having a human in the loop comes at a cost of human errors. And it's really easy to make a mistake, especially if you do the same operation on the daily basis. It becomes so automated that eventually you stop paying that much attention to the details, and then suddenly you realize, it's like cooking. You don't remember, did you add salt or not? And then it's already important to know, because you don't want to oversalt your dish. So and this is where, of course, automation can come in handy. I have seen now papers appearing where people really try to apply it at the scale of the whole lab. So then it's like in the lab, you have a big cube with lots of machines installed. And then eventually there's like a robotic arm, for instance, that performs the experiments. There has been a lot of progress on the basis of individual tools. So, like a lot of those tools are already parallelized when you collect a particular characteristic of your protein, or they are automated that eventually you just like sit at the computer, you set up you just load the sample, and then you like on the computer, you say like what you want to do with the sample, and then everything is done automatically. And then in our lab, for instance, we're doing a lot of microfluidics. This is also like a very promising direction to really collect a lot of data. When you operate in small, on the very small scale. And there it's also like you just generate a lot of data sets. If you, for instance, collect some images of the chip, and then it might be like thousands of images. And of course, processing them manually takes a lot of time. So then it's like, now I have seen a lot of effort to apply automation on those individual steps of data collection, data processing, and then maybe planning of the experiments and combining them all. But of course, we can extrapolate that like within a few years, we should see like completely automated pipelines from scratch. So that will be really promising. On the other hand, you will still, I'm afraid, for now, need like human supervision. simply because it will take some time before we make it like error proofed.
Do you see some key challenges in overcoming this barrier that we need to overcome? Like, what especially is error prone in this procedure in order to commoditize, like automated screening and the machine learning in the back of it that then redesigns proteins? What do we need to productize this? And at some point to have, I don't know, kind of a benchtop device that you put somewhere and you just tell it, yeah, give me the optimal protein for this and that purpose.
So I think that nowadays, primarily, the major challenge comes from the lack of some general tools, machine learning tools. Yeah. So you have specialized tools, for instance. So you have, and then your workflow has to be split into modules, and in each module you might benefit from this like shortcuts that machine learning, for instance, provides, or like this artificial intelligence that it puts in. But you still have this programming effort of putting everything together, making sure that all the coordination is done appropriately, that like the signal is not lost, that like all the parts are basically synchronized, well synchronized. And this is somehow akin to like regular programming experience, when you need to write some protocol for a particular machine, and then it takes a few loops of this debugging, understanding what can possibly go wrong. And this is another point, that like what can possibly go wrong, because unfortunately, even in programming, where we think that programs are predictable, right? Because we write them and like we can see what they do. Even then, it's really almost impossible to have like all these specifications like at the beginning of your project. When you start working on the new data, you always say like, well, let's start working and then we will see what kind of issues can come up. And I think machine learning makes this problem even more at the top of our agenda, because on top of this regular all the possible things that can disrupt your experiment, that like someone accidentally pushes the machine and now it's in a slightly different location, or the sample is loaded in an incorrect well, or like all those things, or sometimes like proteins denature. There is always this extra component now from like machine learning perspective, that you get some accuracy, but it's always like not 100% accurate. So you will always have this gray area where you need your model to be somehow safe and verifiable. And I know that in computer science, it's a big topic nowadays. When people try to come up with some verification of like complex models, or some guarantees that, okay, this model will not venture into this dangerous area because the lab equipment gets destroyed, and that happens also. Like we had some experience with my colleagues in the lab, that like robotic arm goes crazy, starts like pinching holes where there are no wells, and then eventually it's like a mess, and then you have to basically restart everything.
That's interesting, because on the other hand, I've found that some machine learning technologies make programs more reliable. For instance, especially in robotics, there's all these new results coming out where you can, in human language, instruct a robot to do a certain thing. And it doesn't matter so much what language you use exactly, as long as it's approximately the right thing that you described. The robot will do what you intended it to do, which is in a way more reliable than having to exactly input in a certain syntax and know everything beforehand and think about how the robot program would work. But you're absolutely right. I think we get both, definitely. Some more reliability because the user interface is just adapts to you. But then on the other hand, you gain unreliability because, well, maybe you want to exactly specify what has to happen.
Yes, and sometimes you're interested in those really hard constraints, that you don't want to go into a specific part of the parameter space, for instance, because it will cause some troubles. But with machine learning, you then need to somehow incorporate those constraints at the level of the architecture that might be tricky already. Yeah. Whereas in the program, quite often it's just like one extra condition, that you add that, okay, if I'm in those particular specifications, then just like abort the mission.
Absolutely. Yes. Yeah. Okay. So maybe to shift gears even further, the theme of Machines and Molecules is that the guest brings a third M word. Did you bring a third M word?
Yes. Yes, I did. And the M word will be mistake.
And why is that?
Yeah, I was thinking about, I was choosing between mistake and mathematics, because mathematics is close to my heart. And then I figured that probably mathematics will be a little bit boring and some people might not be able to relate to that, but everyone can relate to mistakes. And then it's like, why not? Let's discuss mistakes. I think that it might be undervalued concept in science. Primarily because it's really difficult to imagine an article where people would say like, yeah, I created this protein, I introduced those 3 mutations, and the first 2 were like a huge mistake. I really shouldn't have done that because they disrupted this function. Yeah. So then somehow we tend to report the findings in this like positive way. Yeah. That is like out of 3 mutations, 1 worked. And then readers have to guess what happened to the remaining 2 mutations. So that's why I figured that might be like an interesting topic to discuss. We also, I think, like when we are brought up, there is this culture of like mistake avoidance, that your parents punish you when a child makes a mistake. In school also, like you get penalized, for like making mistakes, which is funny because like when you start doing research, like mistakes is all what you do. Like 99% of the time, your things don't work out. Your hypotheses are incorrect because you forgot to take something important into account, or you put some priorities incorrectly, or you made a mistake in the like experiments. But because of, I think, this discouragement, like from the early age, we are somehow, we tend to avoid this concept.
And what's your approach to mistakes then? How because you say from the early age we were conditioned to avoid them, but now you seem to think they're not that bad. So
I embrace them. No, I try to like also encourage my students, for instance, when they make a mistake, to get accustomed to the idea that it's a normal thing. And what you want to come up with is a contingency plan. Like, first of all, you need to set up the procedures to make sure that you avoid those like mistakes that are important. Yeah. And this is something that we are also not taught, for instance, in school. Yeah. You just get penalized. You get like points deducted if you make a mistake, but your teacher doesn't come up with a plan how to make sure that in the future you avoid this. They sort of assume that like, well, you'll learn somehow. But for instance, in programming, we know that it's almost impossible to write a code without a single error, like a single mistake that like you will run and then it works exactly how you intended it to. And you just learn how to make some checks, cross checks, how to understand parts of your code, for instance, part of the components, and then test them, how to double check the output already, like, does it make sense? Can I somehow do cross-linking with something else and like understand like from a different perspective, will it make sense? So I think this is an ideal strategy how to cope with mistakes that also eventually you try to just double check what you do. However, I haven't seen a good guidebook. So if you like textbook, the 100 simple rules of like avoiding mistakes. So if you have any tips.
No, I don't. But I would. I would take you to a philosophical level as well. I think one of my guiding principles is to learn as much as possible, which includes mistakes. And also, I think what you describe is very compatible with that, because trying to avoid mistakes means, okay, I'm trying really hard to find out where my system still breaks and where I can still learn the most. And The thing is, if you're trying to learn a lot, you never feel bad because you tend to always learn from almost anything that you do in life.
Yeah, absolutely. And in general, I think with the mistakes, quite often they lead to some interesting discoveries eventually. Yeah, there have been quite a few cases when people like by mistake they produce something completely with completely opposite effect, and then suddenly this can be used in like post-it notes or like a glue or something. Yeah, like all those, or like an antibiotics. So all those things. I think like owning up to your mistakes and then testing the limits is a great approach. I also think that in general there is like a spectrum of people's personalities. There are some people who really tend to like avoid making mistakes, so they make a lot of effort to prepare, to do the homework first, to make sure that when they do something, then it's efficient and it's errorless. And then another extreme is more like learning by doing. You just start doing, you make lots of mistakes, but then you learn very fast because you realize like, well, okay, I shouldn't go here and I shouldn't go there. I see it a lot among my friends. Interestingly, I think that given that there are those like two extremes still present, it means that there is no winning strategy, that basically both strategies have advantages and disadvantages. Yeah, eventually.
I guess so. Super, Stas. Thank you for the chat. Thank you for giving a lot of very interesting insight to machine learners and biochemists on the other side.
Thank you for calling me for this job.
I hope your applications for the research grants will go through.
Thank you. It's also like another point of making mistakes and then just resubmitting and scientist life.
Yes. Oh yeah, definitely. Super, Stas. Thank you, and see you at the next conference. Looking forward to it.
Thank you. Me too. Looking forward to it. Bye.
Bye-bye.